Okay, so last time we went over our transcription process. We went over how our introns and our exons differ, where the exons are going to be the part that gets coded and turned into an amino acid, and how the introns get cut out. So when they get cut out, we are going to go through this process called alternative RNA splicing. And what we're doing is we are splicing out our introns. And in this process, we're going to use spliceosomes. And all the spliceosome is pretty much like a protein, and it's going to be used to take out our intron. So here is in the same colors as the last picture, guys. The exons are the darker red color, and the introns are kind of that peach color. The spliceosome is going to attach to our RNA molecule. Now, it's going to bind to the ends of the intron. So the spliceosome is going to bond to the end of the intron. And what it's going to do is it's going to cut that intron out. After the intron is cut out, our exon, if you look over here, we got exon 1 and exon 2. They are going to come together and they are going to join, making our RNA once again complete. There's also another protein called a ribozyme. They're pretty much RNA molecules that function as enzymes. And that's pretty much all you guys really need to know about ribozymes. They're more like proteins than RNA molecules. All right. So that's it for transcription. Now we're going to get into translation. And translation is our process, as we talked about earlier, from going from mRNA to our amino acid or our polypeptide or our protein sequence. Okay, so a bunch of different ways we can say that. So we're going to have our mRNA that just got formed from transcription. We're still going to have our GCAP. We're still going to have our poly A tail. And our mRNA is going to be free of all of our introns. They've already been spliced out. So we have our mRNA that is the coding part left over. So now we're going to go to the protein. So the mRNA that we formed is called mRNA, or messenger RNA. It carries the message from DNA to make our amino acid. We're going to have another mRNA, and this is called tRNA, or transfer RNA. And this RNA is going to help to bring our amino acid over to us. So our mRNA is going to look kind of something like this. We're going to have, it looks kind of like a, I guess, a clover thing here. All right. And we're going to have a bunch of different RNA nucleotides throughout this whole thing. Okay. And I'm not going to draw all of them, but U, G, G, A, okay, and so on and so forth. On the other side of this, right here, that is going to be our amino acid. And this whole thing right here, that is our tRNA. So what's going on here, guys, is our tRNA, in that kind of clover shape, whatever you want to call it, what it does is it transfers our amino acid. Now, all of our tRNAs that we're going to have are going to carry a specific amino acid. All right. Now, I'm going to highlight this area right here on our tRNA, right there. And you guys know that all throughout here, we're going to have a bunch of nucleotides. So just keep that in mind. But I'm only highlighting these three right here, and we're going to come back to them later on. So again, guys, translation, we're going from mRNA to the polypeptide. Now this process is going to happen, as it shows here, on the ribosome. This here in red, that is our mRNA. The mRNA has our codons to it. And what that is, codons, that's our 3 mRNA nucleotide sequences. All right. Now, bonding to our mRNA, and I'm just going to come over here and I'm going to make a short sequence. This is going to be our mRNA I'm going to do in red. So let's say we have U, G, A. And again, this is our mRNA. What we're going to do is we are going to bring our tRNA over. I'll do it blue because that's kind of a blue color there. On that clover area of our tRNA. So I'm going to draw this a little bit differently than the book. In this area right here where I talked about those three, where the U is, there's going to be an A. Where the G is, there's going to be a C. Where the A is, there's going to be a U. All right? And that little area right there is going to be able to fit or bind to our mRNA, and this whole brown thing in the background is a ribosome. On the other side of this, let's use black, is going to be our amino acid. Now, I'm going to go back to a previous slide here. If we go all the way up top, kind of where we had this one here, all right, let's determine what amino acid is going to be on there. Well, we have UGA, mRNA, UGA. We're going to do UGA, and if you look there, it's this little black dot. Okay, let's go to the other one, UGA. What is it? Oh, it's a stop codon. So that's going to be actually the termination at the end of our sequence. Okay. So this right here would be our stop codon. Okay. So that is the one that's going to be on our tRNA all the time for that. It's all for UGA, it's always going to be carried over by the mRNA that is ACU, and it'll always have that amino acid attached to it. So that is our tRNA. Now our mRNA is called the codon. Our tRNA, those three nucleotides, that is called the anticodon. Okay, and the codon and the anticodon, they bond together. All right, so we kind of already talked about this one, guys. The tRNA, it's going to be a particular mRNA codon for the specific amino acid. All right, it's always going to be very particular and match up the mRNA to a specific amino acid. All right, and that's why we have that chart here. And then we already talked about the second one, how the tRNA is going to have the amino acid on the other side of that anticodon triplet code. All right. Okay, so we're good there. There's a picture of the clover anticodon, and up here is going to be our attachment site for the mRNA. Okay. All right, so we are going to have translation occur. Now, when we're doing this, it's just going to be like the other transcription guys, where we're going to have some steps in between everything. The first one and the second one, so on our codon, where you have like our first base pair and our second base pair, they are going to be very specific. Now, our third one is going to be a little bit different. We call this wobble. I'm not talking about the dance. But what this last one is, this wobble, is we might have a different pairing, and it's really not a big deal. Why it's not a big deal is because that third one will probably, sometimes, still go to the same amino acid, where it's not going to change our amino acid. So there is some flexibility in that third one if we have a mutation or a different base pair attached, where it's still going to go to the amino acid. And you guys saw that in the chart that there's like, we only have 20 amino acids and we have 64 different codons we can choose from. Some of them codons are going to go to the same amino acid. Oops. All right, so we're good here. Okay, so on our mRNA, it's going to come out of the nucleus and it's going to go to the ribosome. Now the ribosome is going to be made out of our RNA, or ribosomal RNA. And that's pretty much it. It's a big collection of our RNAs. And on that rRNA, so that's the brown thing in the background, our ribosome, we are going to have 3 different sites. And like I said, guys, it's just going to be kind of the same thing as transcription. We're going to have some steps in there. For translation, we have different positions for our steps. So there's 3 sites, and you guys are going to want to read this kind of like you do regular, sentences. We are going to read this the opposite way. So if you look down here at the picture, it's A, P, and E. So we read it from right to left instead of left to right. The E actually stands for our exit site, so it's going to be our last step. So the A site is actually our first step in it. So what we're going to do, guys, is let me go over the sites with you. The book presents them P, A, and E. I don't know why they do it, but let's start with the A. So the A is going to be our first step with our translation process on the ribosome. So A is going to be our first step, and it's going to make sure that the correct anticodon matches the codon. So in other words, A is going to do this, guys. I'm going to do the mRNA once again in red. So let's just do A, G, G. So it's going to make sure that A, tRNA is going to be blue again. A is going to go to U, G is going to go to C, G is once again going to go to C. If we have something weird where the G is going to A, the A site is going to recognize, hey, something's wrong here, okay? So the A site is like the confirmation, hey, let's make sure everything is cool. The P site is going to be our next step. This is where we are going to grow our polypeptide chain. So the amino acid is going to get carried over to the second site, and then it's going to be pushed onto the growing amino acid chain. So we're forming a polypeptide at the P site. And then lastly, the E site. This is where the tRNA no longer has the amino acid onto it because the amino acid left at the P site. The E site, the tRNA is going to exit. It's going to leave the ribosome, again, leaving that amino acid at the P site to be latched on to the growing polypeptide chain. So once again, we have initiation, elongation, and termination, just like we did for transcription. During the initiation, we're bringing over the mRNA. We are going to bond it to the tRNA, and then hopefully we will have a match there. Okay. Usually, the first amino acid is going to be methionine, M-E-T, and that's our start codon. It's AUG. So here's our picture of the ribosome in the background. We have the A site, the P site, which is blocked off, and the E site. So once we have a good conformation between the codon and the anticodon, the A tRNA is going to move over to the P site. And that's going to start our polypeptide chain. And I told you guys that we usually start off with our start codon, which is methionine. And there's our MET right there. So the start codon starts our sequence, and then we're going to bring these proteins over called initiation factors, and that's going to bring all of our different components of translation together. It's going to bring our mRNA over to the different sites. It's going to bring our tRNA, making sure it binds to the mRNA and brings it over to the various sites as well. So now, after that, we are going to elongate our polypeptide chain. So as we're elongating it, we are going to add our polypeptides to that P site. So the P site is where we are going to grow our polypeptide chain. And again, it's going to go from a 5' to 3' direction for the mRNA, right? So here is everything, guys, and you can see that, here's like a cycle. You can see that as we start to add these on, we are adding them onto the P site, and it's going to keep growing and growing and getting bigger, the different shapes there that are kind of in the bluish purple color. And then after we add it on to the P site, the amino acid, the tRNA, if you look here, it looks like a stick of dynamite. It no longer has that amino acid on it. It dropped it off the P site. And then at that E site, it's going to leave. It's going to go get another amino acid. So once again, it can take that amino acid to wherever it needs to go on another mRNA. So after this, we are going to get a stop codon somewhere. And you guys already saw a stop codon. But that's going to be the end of our mRNA. So once a stop codon reaches the A site, so the first site, we are going to trigger this release factor. We're going to need some water along with that, and then that is going to be the end of our polypeptide chain. And that's going to be where disassembly occurs. So release factor comes in, and now we got our free polypeptide, and it can go to wherever it needs to go. So after this, we are still going to modify our polypeptide chain a little bit. And we learned about this in a previous chapter. We have primary, secondary, tertiary, and quaternary different shapes of our proteins. So after we have our polypeptide chain, we are going to change it from being two-dimensional to a three-dimensional shape. And just think about enzymes, guys. Remember enzymes? We had that like active site where the substrate can bond. That active site was just part of the enzyme, and that whole enzyme is a three-dimensional shape. If we just have a long polypeptide chain, it's not going to do anything for us. It's not going to have an active site or anything like that when it comes to proteins. So we make it into a three-dimensional shape. We do some hydrogen bonding between the R groups of our amino acids, and that helps us to get our three-dimensional shape. So our last thing we're going to go over today is just the locations where we want our polypeptides to end up. Now don't forget, guys, we have ribosomes in two different areas in our cells. We have free ribosomes just floating around the cytosol, and then we also have bound ribosomes that are going to be attached to our rough ER. So free ribosomes, they're mostly going to make proteins that are going to be used inside the cell. In the rough ER, most of the time it's going to make proteins that are going to be exported out of the cell through the cell membrane. So regardless of where it's going to go, outside the cell or stay in the cell, it's still going to all start in the cytosol, this making of the polypeptide. It might stay in the cytosol, or it might go to the ER. And determining where it's going to go after the original protein is made, that's where we are going to bring in something called a signal peptide. So we're going to have a signal recognition particle, an SRP, and it's going to bind to this signal peptide. And that's going to determine, hey, is it going to just stay in the cell? Is it done? Is it made on the free ribosome and everything is good? Or is it going to go to the ER? And here you can see that everything is going to attach to that polypeptide after it is made. And if that signal peptide is attached, it's going to determine where it's going to go next. All right. And then after, if it goes to the ER, then if you remember, it goes to the vesicle, to the Golgi, gets marked, and then it leaves the cell. If it's going to stay in the cell, then it just stays in the cell, gets used, and the cell tells it where it needs to go. Okay, so I think that's enough for today, guys. Next time we will finish the chapter. Have a good rest of your day.